Good morning, students. This is your teacher Neela Melawat, and today we'll start with our practicals. I know, students, we are in the lockdown and we are unable to come to the classroom, but I'll make sure that nothing will stop us from studying, nor the lockdown, nor the coronavirus. But it is equally important that you should come forward and study hard. Okay, dear students? So I hope we all are together, isn't it? Okay, so today we'll start with the experiment number 1, and I hope you enjoy this experiment. Now we know, students, we've talked about pollination. What is pollination? Transfer of pollen grains from one flower to the another. And we know that insects, or you can say the biotic components, they play a very important role in the pollination. Insects especially, they visit from one flower to the another and they help in the transport of pollen grains. Isn't it, students? So flowers beautifully colored, the insects, they'll be visiting the flowers for nectar or for staying, right? And for different awards, for getting different awards, and in turn they will be transferring the pollen grains from one flower to the another. Right? The flowers, they are beautifully colored, isn't it? And that color attracts the insects. And a flower, when this pollen grain basically reaches to the pollen stigma of the ovary, it then forms a pollen tube and slowly it reaches to the ovary. Right? This will bring about the fertilization process. So in a way, I can say that the biotic components, they play a very important role in bringing about the fertilization. Because if they are not there, then the chances of being transferred, the pollen grain being transferred to the stigma, they are very, very less. Okay, they basically increase the chances so that maximum number of pollen grains they reach to the flowers, and that will result in pollination, and ultimately they will lead to fertilization also. And right, now we know pollinating agents, as I was describing, there are two types of biotic agents. One is abiotic, another is biotic. Wind and water play a very important role in transfer of pollen grains. Same here, biotic components, the birds and insects, they help in the transport of pollen grains. Right, you see here it is being transferred there. The birds, the hummingbird, the wasp, they will be visiting the flowers and they will transfer the pollen grains to the female part. Then there is a fertilization process will occur, pollen tube formation will occur. The pollen tube, when the pollen tube formation occurs, it basically has two male gametes, and these one of these male gametes will fuse with the egg, resulting in the formation of the zygote, while the another cell will fuse with the polar nuclei. So that is why this process will bring about the double fertilization. Okay, dear students, this process will bring about the double fertilization. I hope you remember this. So double fertilization is there. The very first step is called the syngamy and the second step is called triple fusion. So today we'll be performing an experiment. We will see the formation of pollen tubes. Okay, how pollen tubes are formed, how pollen germination occurs. So we will perform this experiment. So our aim of the experiment is to observe the formation of pollen tube. Okay. Now for this experiment, for this activity, we'll require certain materials, and the materials are, we'll require a flower, we'll have a distilled water that is present in the beaker. Okay. Then we have 30 grams of magnesium sulfate, 10 grams of boric acid, 20 grams of potassium nitrate. Okay, dear students, and 10 grams of sucrose. Sucrose is your sugar. Okay, so we will have distilled water, we will take a flour, 30 milligrams of magnesium sulfate, 10 milligrams of boric acid, 20 milligrams of potassium nitrate, and 10 grams of sucrose. Right, we will use the dropper, glass rod, then slide. Okay, clear everyone? We will use the following materials. Now the very first step that we'll do, First of all, we'll add all the 4 salts in the distilled water. So we will prepare a solution that is called a mineral solution. Okay, so we will mix all the 4 salts that we have weighed and we will make a mineral solution, a nutrient medium we will produce. Okay, we will make a nutrient medium. Okay, we will mix all the 4 salts in distilled water and we will make a nutrient medium. And while we are mixing it, we'll thoroughly use the glass rod to mix all the salts. Okay, so to prepare the nutrient solution, we will add all the 4 salts and we'll use the glass rod so that all these salts, they are properly mixed in the distilled water and our mineral solution is prepared. Our nutrient medium is prepared. Now we'll take few drops of this nutrient medium and we'll put it on the slide. Okay, हमने dropper लेना है. Dropper में कुछ drops हमने इस solution की लेनी है and we'll put few drops on the center of a slide. Dear students, the slide should be properly clean and dry and the dropper should also be properly washed. Then we will use few drops of this nutrient medium and we'll put the drops on the slide. Okay, now we'll take the flour and we'll dust a few pollen grains on the slide. Okay, we will drop some pollen grains on the slide. Clear, students? See, this is the structure which will be there. We have few drops of the nutrient medium on the slide and we have put few pollen grains into it. Okay, dear students, we have dusted some pollen grains into the solution and with the help of the pen, with the help of the needle, we will place the cover slip over it. Okay, we will place the cover slip over. We will cover this with the help of a cover slip and make sure while we are placing the cover slip, there should be no formation of air bubbles. Okay, so from all sides, the cover slip should be properly dipped into the solution and very slowly we will put it on the slide. Now our slide is ready. Okay, dear students, the slide is ready. We will keep it for 2 hours. You can leave it for 30 minutes to 2 hours. And what you will observe slowly, when you put it under the microscope, adjust your microscope first under the low microscope, then under the high power. So first we'll see under the 10x, then we'll shift it for the 40x and we will observe. So what we'll observe? This type of pollen grains will be visible under the microscope and you can easily locate the nucleus and the generative cell. Okay, dear students, you can easily locate the generative cell. Now after some time we will see a small tube has started. There is a formation of small tube from the pollen grains and that small tube is nothing but the pollen tube. Okay, and after a few hours again you will observe that the pollen tube has continued elongation, right? It is continuously being elongated and you can easily locate 2 male gametes here. You can easily locate the male gametes. 2 male gametes will be visible to us, right? The 2 male gametes will be visible. We all know that there are when there is a formation of pollen tube, then there are 2 male gametes that are being transferred. And we remember that 60% of the pollen grains, they are shed in the 2 cell stage. Okay, right? In that stage, one cell will be there. One is generative cell, other is vegetative cell. Then the generative cell will divide to form the 2 male gametes. Okay, and in some cases the pollen grains, they are shed at 3 cell stage. Okay, so 2 male gametes are there and we can easily locate them in the tube in the starting. So thus we will observe after 2 hours, we will observe that there is the formation of pollen tube has started in the pollen grains. Okay, so this is how we can easily observe that the process of pollination occurs when the nutrient medium is provided. The same medium is provided in nature by the stigma of the ovary. Okay, the nutrient that we are giving in the lab is artificial, but the nutrient in nature is provided by the stigma and that will lead to the growth of the pollen tube. Okay, dear students, So I hope you have enjoyed this video. Okay, so if you've liked the video, please do like the videos, please do subscribe the channel, and this will really motivate me. Okay, dear students? Okay, so yeah, now coming to your homework, you will make notes of this practical. Whenever we'll meet students after the lockdown, we'll definitely perform this experiment in the lab. Till then, you can collect the information, you can understand how this experiment can be performed. Okay, then you will be collecting the samples of different flowers and will perform this experiment in the lab. Till then, just enjoy it hypothetically. Okay? Okay, bye everyone.